First, it's a great pleasure to introduce Nada Jabdo, our keynote speaker for the EMBO workshop, Physiology and Function of Histone Variants. So we welcome our meeting participants, as well as other attendees who are here today for this open lecture. And we thank Nada for allowing us to open it up to the public. Nada is a professor of pediatrics at McGill University and a physician at the Montreal Children's Hospital. As many of you know, Nada treats pediatric glioma patients, and her lab is hugely invested in understanding the role of oncohistones in pediatric cancers. She's a true pioneer in this field and made the discovery of H3.3 mutations in pediatric GBM, and has made significant strides in understanding the role of these mutations, cooperation with other driver events, and importantly, how they affect the epigenome. As many of you know, her energy and enthusiasm is truly inspiring, and her lab is truly dedicated to understand how we can therapeutically target these oncohistones. Nada leads local and national projects to provide not only NGS data for pediatric cancer patients and also, again, working towards therapeutic targets. She's funded by the National Institutes of Health, the United States, and the Canadian Institutes of Health Research, and was also a member of a Stand Up to Cancer for Pediatric Brain Tumors. She's a fellow of the Royal Society of Canada, the 2020 winner of the Dr. Chu Wai Memorial Prize in Cancer Research of the University of British Columbia in Canada, and recently won the Canadian Cancer Society's 2020 Robert L. Nobel Prize. So we welcome you here today, Nada. Thank you so much. Just a reminder for the meeting attendees that this is a single Zoom link. We must return to the auditorium to start the next session with a different Zoom link. So please remember to do that after the talk. And Nada, please take it away.
Thank you very, very much, Marcus and Emily, for holding strong. It has been like a challenge for this meeting, and this is such an important meeting. It's really, I feel there is a character in French cartoons, Obélix. He fell into the magic potion and became whatever. I feel like I fell in the magic potion of histone variants, and this is such an inspiring community and such an interesting things. And there are so many things that we are kind of learning as we're going forward. My ultimate aim is to try and help those children that I treat that die, children and young adults actually. And again, it's by understanding that you can help better. And I hope that I tried in this keynote, and thank you very much for giving me this honor of providing a keynote at this meeting. I tried to put things that were already published, things that were just accepted, and new things that really are in the making, so that to make it a little bit more kind of interesting. I have no disclosures except that, you know, gather is my first thing, and I never learned something. I'm stalking. I really never stalked anyone, and I'm now enjoying stalking, and I'm stalking Marcus. So Marcus, beware, I'm stalking you wherever. This is my only way of finding my links to where I go, and thank you again. So the work that we've done and the histone mutation that we've uncovered, the first oncohistone, because now there are so many more, and you've heard Laura talk about the neurodevelopmental issues with histone mutants, started with our discovery in 2012, the discovery that I chair with a group of Susie Baker of recurrent hotspots, somatic mutation on specific residues of histone 3 variants. The lysine 27 that you know can be monodi and trimethylated and also methylated gets changed to methionine and in rare cases to an isoleucine. The glycine 34 that doesn't get post-translationally modified exquisitely and exclusively up to now on histone 3.3 gets changed to an arginine, a valine, or a tryptophan. And the lysine 36 that also gets monodi and trimethylated by multiple distinct methyltransferases gets changed, similar to the lysine 27, to a methionine, and in rare cases, an isoleucine. So again, those mutations are somatic. They're heterozygous. They barely account for 10% of the total histone 3 pool whenever they're present in the cell. And the K2M mutation, whether K27M or K36M, have a really dominant gain of function effect that we really beg understanding. The other thing that I would like to bring about as early as now is that when you have K27M, the K27 methylation and the lysine 36 methylation have been known to be opposed in mark. meaning there is really limited overlap between di and trimethylation of lysine 36 and di and trimethylation of lysine 27, and especially in the trimethylation component. Those really mark distinct boundary, and it so happens that those mutations are occurring and are mutually exclusive with each other in those residues. And why is that? Let's start first with the K2M mutation. We've uncovered K27M to actually specify the brain midline in kids. And actually there is super tight link with brain development, because based on the age and the location, you start in the pons, you go to the thalamus, and then it's really the midline brain that have those K27M mutation. It's exquisitely linked with brain development, and the associated mutation are co-opted based on their role in those developing areas of the brain. More than that, the new oncohistogenic that was uncovered by the group of Marcel Kuhl and David Allison, EZH interacting protein, actually specify the midline tumor that are wild type for K27M. And really, the brain midline in gliomas from the age of 3 years until the age of 50 years is specified by K27M or oncohistone K-EZ. And in the other place where you find those K27M mutation is actually AML. And more than that, in myelodysplastic syndrome that will go into AML, you find about 7% of K27M mutation in any of those histone 3 variants. And we've identified in head and neck cancer, in about 15% of cases, either NSD1 truncating mutation, K36M mutation that also occur, or any of their histone 3 variants. And the group of Adrian Flanagan and Peter Campbell in Oxford identified in chondroblastoma lysine 36M mutation that specify 96% of those presumably benign but very maiming tumors. And last, we identified in the in undifferentiated sarcomas about 20% lysine 36M mutation in histone 3.1. So that is to say that when you dig, you find, and in some instances, the K36M and the K27M mutation really specify those entities, and they never coexist with each other. So how do they promote cancer? And this is where the first hint came from the beautiful work of Peter Lewis, that was a postdoc at that time in Dave Ellis's lab, where he did what he called the alphabet experiment. And what he did also is he's changed lysine to methionine in any of the 4, 9, 27, and 36 residue in this histone 3 tail, where you had a lysine, and he changed it to methionine. And each time he changed it, what he saw was a drastic decrease in the total level of the methylation of the respective residue. Remember, this is really, those mutations account for barely 10% of the total histone 3 pool. However, the effect is far from, if the person in the Institut Joseph could mute their phone, that would be very appreciated. So if those mutations lead to a drastic gain of function and a drastic depletion of the methylation of the respective residue. And people had initially thought that they are actually inhibitor of the methyltransferases, and that K27M by extension was inhibiting EZH1 and EZH2. So Chow in Dave Ellis's lab, and this is a work that was co-led by Dave, Peter, and me, showed exquisitely the first thing, that those mutations are oncogenic and they block differentiation. Because the K36M was the only mutation that was identified in tumor, whereas in K27M it was a nightmare with other associated mutation, he started in that scenario and used it in mesenchymal stem cells, where those tumors seems to be, originating from. And when he put the K36M mutation, whether in histone 3.1, 2, or 3, what he found was a blockade in differentiation. Those mesenchymal stem cells could not differentiate, and they became actually tumorigenic because they generated undifferentiated high-grade and aggressive sarcoma when put in the flank of nude mice. And when he looked at the epigenome, this is the first thing that he saw, where you have here in the wild type, the lysine 36 trimethylation pasting gene bodies as expected, the lysine 36 di, which is quite an abundant mark, pasting other intergenic area, and mutually exclusive or barely any overlap with the lysine 27 tri, up in addition of the K36M in those mesenchymal stem cells. As expected from the work of Peter, we saw a drastic decrease of those tri and dimethylation mark on lysine 36. And what we saw was actually gained from the boundary where you had K27 trimethylation. We saw this mark in the presence of K36M spreading to areas that were devoid of K36 di and K36 tri. And what was the consequence of that? If you look, this is K27 tri and the wild type and lysine 36M. The aim of K27 tri is to recruit the PRC1 and to mediate the level of repression and compaction of DNA. So what happened in the context of K36M, the gain of K27 tri in areas that were devoid of lysine 36 tri and di led to a redistribution, a dilution of ring 1b and of PRC1. So because everything is in rate limiting quantities, when you had a mark that went and spread more, the repressive mark that recognized the reader of that mark became diluted in its effect, and actually in up down ring 1b, we could mimic the effect of lysine 36M in those mice. So that led us to propose this first model where usually you have your genes that are turned off because they're pasted with K27 trimethylation, and PRC mediates the compaction and the silencing of this. And in the intergenome, you have area where you have compaction and silencing, an area that have lysine 36 di that are more active and they are doing other things. When you have K36M or NSD1 loss, actually, and this is what we showed, actually lose lysine 36 di. This allows undue spread of the K27 tri in those intergenic regions, and this leads tension to a dilution of the repression of PRC1, and this leads to a block in differentiation because you repress things that should not be repressed and express potentially stemness gene or progenitor genes that should have been completely silenced, and this actually promotes and fosters sarcoma development. So what about K27M? Because this is the one I'm super interested in. Remember, I told you that the initial work was saying that maybe those K2M mutation are acting as inhibitors of the metal transferases. This is true because you see a drastic depletion, but those are not real EZH2 inhibition. Because if you have EZH2 inhibitor like the pathology, you really completely remove any, to the same extent, monodi and trimethylation. And what we saw exquisitely, and this was a conundrum that was very hard to reconcile, that in the case of K27M, this is a tumor that's wild type, and you see a lot of trimethylation of lysine 27. If you do immunohistochemistry on a mutant K27M, you say, oh my God, it's completely lost. Well, no. There are areas in the genome that actually have residual K27me3. And this residual K27me3 became the subject of a lot of controversy and a lot of issues. And I'm going to go over some. I think the jury is not out on that, but our data is, I believe, quite strong, and I hope that I will convince you that we are on the right track. It's not that the others weren't, it's just that the use of isogenic model system in the cell of origin where those mutations occur is a major in these and allowed us potentially to tease better out the effect of K27M. So the first hypothesis came because of the high affinity of the K27M to the metal transferase domain is the sequestration of PRC2 to specific areas. This would lead to kind of this residual K27me3. Great. The other hypothesis was brought by the group of Christian Helen, and he thought that actually high affinity site for PRC2 would still be able to recruit the PRC2 complex despite the presence of K27M and mediate the position of this mark. And unfortunately, undue suppression, like for example, for the INC4A locus, and that would be the reason why they lose those tumor suppressor and they become oncogenic. And actually, the use of EZH2 inhibitor in that case, and he was the first one to show it exquisitely, would be beneficial because it would alleviate this suppression and would potentially restore breaks that were completely gone. The third hypothesis came from the group of Ali Shilatifar, where he said, okay, K27M cannot have K27 tri, and in the nucleosome, you have two histone 3 variants. So the other one becomes acetylation, and we have, in fact, increase in K27 acetylation in the context of K27M. This leads to a new oncogenic enhancer landscape and repulses PRC2 away from these areas, and wherever you don't have K27M, you could potentially land PRC2 and deposit those K27me3. So, okay, great hypothesis. But I think what is happening, and we showed it in multiple isogenic lines, and actually the work that was led by Peter Lewis on EGH and the interactive protein, which is explicitly the same thing, further confirmed. EZIP is not incorporated. It doesn't need to be a nucleosome, still mediate the same type of effect than K27M. And actually what we think is happening, and this work was co-led with Jacek Majewski and Ashot, a postdoc in my lab, Brian, a graduate student in my lab, and Haifet postdoc in Jacek's lab, co-led this work, showed exquisitely that it's one step down. Actually, this enzyme is able to mediate mono to the same level than a dimethylation in a wild type of lysine 27, and dimethylation to the same level of K27 trimethylation, and barely any K27me3. And actually, what we showed exquisitely is that you recruit PRC2 in cell of origin where K27M mutation occur. PRC2 gets recruited normally without any problems with the nucleation site. The problem it has, because it's so slow in depositing, especially the lysine 27 trimethylation, is that within the given cell cycle it has, it's only able to deposit the bareness. So you have restriction of K27me3 to specific CGIs, which are the normal recruitment sites of PRC2 in this cell of origin, in this progenitor. So the hit of PRC2 happens, the one that was described by Danny Weinberg. However, the spread of the K27me3 and a certain level of the Me2 mark are not happening. And you really have only K27me3 within those areas. And I think those areas are humongously compacted and silenced. And this is one of the main issues here. Nothing is void in chromatin. Why is this important? If you look back as what you need to do, if you're a progenitor, in order to come and differentiate, you need to be able to play with your PRC2 and your PRC1, and by extension with your trithorax. In a progenitor cell, in embryonic stem cell is actually the most exquisite part, but by extension in progenitor cell, you have areas that have PRC2 that recruits it and that silence it. Areas where you have patterning gene, the Hox target genes, and PRC2 target gene, and differentiation gene that are sometimes bivalently marked because you want them on very fast to release PRC2 from both sides. And other areas that have this activation mark, those are stemness genes that need to be expressed within the context of this progenitor. Based on cue from the environment and push, your PRC2 that landed will be able to spread and get removed from some area to go to other area to silence. And this is what's not happening in K27M. This mutation just makes the progenitor keep the original image of the K27me3. Only those sites that PRC2 landed in that progenitor are able to maintain it. And your PRC2 is not nimble anymore, cannot move elsewhere, cannot repress other instances. And this is what created, like Peter Pan, it's the progenitor cycling indefinitely in the same perpetual progenitor state, unable to further push into differentiation. And the work of Mario Suva and Mariella Philbin on the single cell shows that actually oligodendroid site progenitors are the cell of presumed origin of K27M. And we show that they cycle perpetually. And when you remove the mutation, we push it to differentiate, we push cells to differentiate and acquire those differentiation, and we release the block. This is something that's reversible. And the other thing, okay, you have restriction of K27me2 and M3, and mainly of M3. This is really the common feature between EZH inhibit interacting protein and K27M. What happens to areas that should have had K27 die? The work of Danny Weinberg showed that there may be an increase in the lysine 36 dimethylation, and we and others showed that there is an increase in the K27 acetylation. I think it all depends on what were the boundaries in the cell of origin. If there were a lot of lysine 36 dye, this lysine 36 dye is there. If there was limited lysine 36 dye, because this progenitor was a little bit further down differentiation pathway, the loss of the lysine 27 dye gets replaced by K27 acetylation, as we exquisitely show in association with Claudia Kleinman and Steve Mack, where we showed pervasive acetylation. And this work was led by Brian and Nick who are graduate students in my lab and actually postdoc in my lab. And they showed that really this acetylation, this increased acetylation wasn't leading to new enhancer and promoter, but was more pervasive within the intergenome and was leading to a little bit of an increased level of endogenous retrovirus expression. Because remember, Histone 3.3 also paced all of those endogenous. This is beautiful work by David Alice, paced those endogenous retroviruses. And the presence of this acetylation, this site, increases their basal expression level. And you could push it further by using DNA demethylating agents that remove the repression on those ERVs that are silenced by DNA methylation and make this tumor potentially a little bit more hot to the immune system. If that's true, we should see it in other model systems. And this is work that was just accepted in molecular stuff. It's been ongoing for a few years. We tried to model it in the Drosophila. The Drosophila is a beautiful model. It doesn't lie. The phenotype is quite striking. And it doesn't need DNA methylation. It has those histone markers are the one that mediate a lot of the things on the genome of the Drosophila. So what we did in collaboration with Paul Lasko and Martin Hasselblad in Münster, Carol and Amel Shaouch in my lab did put the K27M and the K36M in multiple drivers. And we observed similar phenotype. And one of the drivers, which was an eye driver, gave us a very exquisite phenotype. It gave a small eye phenotype, but not only small eye for K27M, it was tumors and very disorganized eye. And it was 100% penetrance. And the lysine 36M gave also this type of phenotype to a lesser extent and with lesser penetrance. And when we look in K27M, as expected, the di and trimethylation of lysine 27 were completely gone, as were actually for lysine 36M, the di and trimethylation on lysine 36. And what we saw is, remember what I told you, K27 di and tri and K36 di and tri cannot exist. Those marks, when they were gone, were replaced by the opposing mark. Lysine 36 di in the context of K27M and lysine 27 di and tri in the context of K36M. What else did we see? We saw actually, when we did the transcriptome on those mutant fly, we saw an increase in pi RNA genes and really a very strong degree of genes. Can somebody mute? Can you mute, please? Do you mind muting your microphone? So we saw the K27me3. We saw the K27me3 getting pasting eye-specific genes and leading their downregulation, and to a lesser extent in K36M. And we saw the loss of those K27me3 in germline and piRNA biogenesis gene. And the other thing that we saw is we saw a huge increase, especially in K36, of expression of transposable element, full families of gypsies and others that were highly expressed. Why is that? When we looked at the epigenome, this is a chromosome, this is representative of the 4 chromosomes in Drosophila. This is a pericentromeric region, this is a chromosomal arm. What you see in the wild type for K27me3 is that you have barely any K27me3 in your pericentromeric areas, and you have all of those across the silence element across the chromosomal arm. In the context of K27M, as expected, you have a huge loss of the K27me3, but some areas unduly keep it, really keep it. And to our surprise, we saw a redistribution in K36M, we saw an increase in the basal K27me3 across the board. But that was made at the expense of other areas that should have been pasted with K27me3 that really were decreased there. The same for K36 di. One of the other surprises that we had is that the K36 di deposition was superbly enriched in the pericentromeric areas. And look what happened in the context of K27M. The redistribution of K36 di to this areas of the chromosomal arm came at the expense of the pericentromeric region that became depleted actually of K36 di. Both in the context, because you inhibit the position of K36 di in the context of K36M, and you redistribute it here. So just to recap, when you have K27M, you lose K27me3, except in specific area. those areas get replaced by lysine 36 dye, which becomes displaced from its pericentromeric regions to the chromosomal arm. And in the context of K36M, the loss of lysine 36 dye leads to a redistribution of your K27me3, which actually loses its inhibition on specific area and gain it on other areas that should not have it. And this is actually what leads to the increased expression, the delta in K36M and K27M of lysine 36 dye is humongous and leads actually to the expression of class 1 and class 2 transposable element, mainly in K36M. Why is that? Because in K27M, you actually have increased expression of the pi RNA gene because the germline that serve to repress those transposable elements, and this is why you don't have it to that same level in K27M. So we tried also doing a rescue screen, and when we did using ASH1 RNAi, ASH1 is the lysine 36 dimethyltransferase, we rescued the K27M and we actually made the phenotype even more severe in K36M, and EZ, which removed K27 di and tri, made it lethal in K27M and rescued K36M. TET2 and SD are not super expressed in the iDisk, and it's really ASH1, the main lysine 36 dimethyltransferase, and this is why we presume they didn't have a lot of effect there. So just to recap this, you have K27M leads to small disorganized tumors eyes. They actually, the eye-specifying genes are repressed, and you have transposable element de-repression and piRNA machinery induction that limits the amount of transposable element de-repression. In K36M, you have eye-specifying genes that become unduly repressed and a huge de-repression of the transposable elements, probably due to the loss of lysine 36 dye in those areas that potentially serve to repress. Remember, the work of Chao Lu in Nature about a year ago showed that the lysine 36 dye serves to recruit the NMT3A and deposit DNA methylation. And maybe this is intergenic methylation that represses the transposable elements in the intergenome. And we're seeing the effect in the Drosophila because they don't need the layer of DNA methylation. So with this, I hope that I've convinced you that in the context of K27M, PRC2 gets recruited to its nucleation site and only to its nucleation site. And this is where K27me3 gets deposited. From boundaries, the lysine 36 dye, lysine 27 acetylation can migrate and actually maybe promote added events, some that could be taken advantage of for therapeutic issues, and others that potentially could promote oncogenesis. And we need to learn more about them. So I'll take the last 5 minutes to kind of speak about the G34 tumors. The G34 tumors are in hemispheric glioma. There's something quite striking. You have actually R and V in the brain, and you have W in the bone. Osteosarcomas and giant cell tumor of the bone have W, and you never have W in the brain, never. It's the same residue, different mutation, different localization. There are people in the work of, we collaborated in this work, Peter Lewis showed that they inhibit ZD2 and NSD2 based, it's a bulky amino acid change that prevent the recognition of lysine 36. And by extension, PRC2 is able now to spread and mediate K27 trimethylation. We work in the giant cell tumor of the bone and we CRISPR'd out the mutation. And what we exquisitely showed is actually you have something that was happening very slowly in 2 steps. The loss of lysine 36 trimethylation and dimethylation maybe led actually to K27me3 going to those areas. And by extension, eviction of histone 3.3, because histone 3.3 mainly live, as shown by others, in active transcription sites. And this leads to genes be getting repressed. And from those boundaries that lost the K27me3, because it got redistributed, we got redistribution of other chromatin mark that probably participate in oncogenesis. And the net effect in those giant cell tumor of the bone is similar to what we see in K27M. Stall differentiation, they're unable to go further. And actually they secrete components, remodel the matrix metalloproteases and lead to bone resorption and the tumor that we see. The glioma, and I'll go fast on this because this is something that we've shown. What we showed is that the G34 mutation in the high-grade glioma is not the high-grade glioma. It occurs in interneuron progenitors. It stalls differentiation and it co-opts using a previously present chromatin architecture, the promoter of PGFRA, that instead of getting bivalently silenced, as it should have been in the ganglionic eminences, where this interneuron, the cell of origin, this tumor arises, usually they are silenced, they get expressed, and PGFR alpha is the major oncogene. So those gliomas are in fact neuronal tumor, probably embryonal neuronal tumors that occur in interneuron progenitors. And PGFR alpha is the main oncogene that co-opt this. And this is something that we could target today. And I want to share very fast some quite exciting things. We always ask ourselves, why arginine, valine, and W? Why are they different? Why those tumors? Why no W in the brain? Because they're not doing the same thing. And CMAC has spent 4 years of her life trying to generate those direct knock-in mice who were successful at last. We can't derive K27M, K36M direct knock-in, but we can derive these before Histone 3.3 knock-in mice that are either mutant for arginine or valine or W instead of a glycine 34. And we have them on 2 distinct backgrounds. It's not a background thing. And look what we have. The W have 0, almost close to 0 neural phenotype. The R have a major neurodegenerative phenotype that I'm going to show. All of them are obese, but the most are the W. And it's not because they eat more. There's something with the adipocyte. And the W have a mesenchymal phenotype with kidney and bladder phenotype. Look at how obese those mice, and we put them in a metabolic chamber. There is something about the fat that there is something that tells the mesenchymal stem cell to go and produce fat way more than others. It's not because they're eating more. And the W, as I said, have bladder phenotype, whereas the brain, they have really a neurodegenerative phenotype. They're fine until the age of 6 to 8 weeks, and then they become ataxic. They have hindbrain clastic. They really show signs of severe neurodegeneration. And they can live longer, but they are really completely hunched and they don't do anything, and have done multiple developmental things. Their brain, similar to what was shown in Laura, they have microcephaly, and they're actually losing specific layers of the brain. They have a very bad cerebellar architecture, and they have increased microglia and astrocytic signature. The R is really completely full of those microglia, whether at the single cell or at the protein level, and they're losing neuronal components. And we're trying to understand, and we have some clues, why R is different from W in the brain. There's something quite unique. And actually, those are tools that are leading us to understand a little bit more what are the specific events that are needed during normal brain development. And when they're missing, what is it that's happening? And I think this is the exciting part. I hope that I'm convincing you that oncohistone in cancer are really a way to stall development. And there's something that happened on the mark, but also there are neighboring marks that take advantage of what can be done, and we need to better understand them in the context of where they originate in. That one mutation is not equal to another, and there's so much specificity. The histone code that Dave Alice all the time speaking about is beautiful. And this work could not have been done without amazing collaboration with Daria Kleinman's group, with Jacek Majewski's group, with people at St. Justine, with Michael Taylor. Amazing collaboration that we're going, the amazing collaborator across the world, and our funding sources. And thank you very much for your attention. Just to say, we're recruiting. If anybody's interested in joining my lab, postdoctoral and PhD positions are available. And thank you very much.
Thank you, Nada, for always a beautiful talk. So much information, so much to digest, and so many new and exciting findings. We have little time for questions, so maybe we'll take 1 or 2. We have some lining up in the Q&A here. I'm going to read them for sake of time. The first is from Yamini Dalal. Hi, Yamini. Not a great talk as always, fascinating insights. Would the lack of H1 variants in melanogaster alter how these key 3.3 mutations might work in human cells, especially glioma?
Yeah, I cannot exclude this. This is something that we should really envisage. Yeah.
Okay. Carolyn Luger asks, I would assume that the G34 mutations affect modification of neighboring residues, i.e. or e.g. K36.
Yes, and this has been shown by Peter Lewis and by other groups. However, I think they're not equal in the way they are affecting, and this is why we're seeing, I'm kind of giving you a little bit of the punchline of our finding. It's this mutation, this is why we're seeing this, those different, they're not equal in the way they affect those residues.
Okay, and just to follow up, one last one from Bagshri Jamanj. When K36 dimethyl is redistributed from repeats to transcribed regions, what happens to K9 methylation?
It doesn't change. I was so surprised by that. We did it. It doesn't, not a single, it's, you know, it's still bulk, but, you know, on the bulk, I was expecting something. It doesn't change.
Okay, and to follow up that, since we're still on the variant discussion here, he also asks, or she asks, do you observe redistribution of variants like H2AZ?
So this is what I would like to see. And again, remember, this is Drosophila. Just getting a chip on Drosophila, we cried. I had my daughters actually work as a summer project, and they hate me. They will never do research anymore. Dissecting fly eyes, you need millions in order to do that. But yes, we need to revisit this part as well.
Yes, very interesting questions and lots to think about. Thank you so much, Nada.
Thank you very much.
It was excellent. And just to remind everybody, the meeting participants, that we will now exit Zoom, return to Gather Town, and we'll need a few minutes on the side of Marcus's team to relink us to the next Zoom link. So we'll be on a new Zoom link. So please, when you exit, just give us a few minutes to enter into the next session. Thanks, everyone.